Okay, this lecture is about the bacterial diversity and the ecology. And this period, I will talk about the ecology more. And then in the next period, I talk about the archaea more, so that we can cover the whole of the both domains, that is both domain bacteria as well as domain archaea. The study of bacteria, before the study of bacteria, let me introduce the evolutionary thinking on the bacteria. There are many studies of how bacteria evolved, and I give you a brief introduction to the thinking process as well as the research on it. We can get fossils from sedimentary rocks, and so they are starting from the large fossils. We call them the Paleolithic, Paleozoic forms, and the 3 stages of the geological age. But bacteria is very small, so how to study it? So scientists, especially geologists, they try to grind up for ancient sedimentary rock and to look for the form similar to bacteria. And biochemists try to extract those material to see if there is any carbon and residual carbon in it, especially the 6-member ring carbon, because it's most likely made by the living material. So about 1950s, scientists started looking for the evolution of bacteria. And the time frame of evolution was set by the astronomical studies, because when people are able to get the meteorite from the moon, and the meteorite may be formed when our solar system is formed. So dating the meteorite is likely to show the length of our solar system. So it was dated to about 4.6 billion years ago. So the Earth now is generally believed to form about 4.6 billion years ago. And the zircon crystal is when temperature of the Earth is cooled down, then the zircon crystal is forming the first or the earliest sedimentary rock. This is about 4.4 billion years ago. So this is that means only about 200 million years for the Earth to cool down, so cool as to form the sedimentary rock. And the scientists are trying to get the fossils out of it. Okay, the oldest rock is found in Greenland, southwestern Greenland, which is dated about 3.86 billion years ago. So it's called the Isaac Geis. And it has some sedimentary rock in it. Other ancient rocks is from the Western Australia or Southern Africa. So the oldest ones are about 3.5 billion years ago. And scientists grind it up into thin sections and watch it under the microscope, and they find some circular form, that is a coccus form, thin or bacillus form. And so they think that maybe the earlier form of bacteria. However, I think that it's not easy to say if it's formed abiotically, that is chemically aggregate, or it is from bacteria. And so scientists is trying to also try to mimic the conditions of early Earth. So by finding out the ancient rock and find out the content of the ancient rock, and scientists are able to show that the ancient rock shows that the early atmosphere is has very low amount of oxygen, but may have other kind of things in the atmosphere, which is methane, carbon dioxide, nitrogen, ammonia, so small amount of carbon monoxide, hydrogen sulfide, cyanide, hydrogen cyanide. And so scientists try to simulate the atmosphere and try to do abiotic evolution, that is chemical evolution, by supplying the thunders, that is electric sparks, by supplying the vapors to its glass container, that is boiling water in an atmosphere which contains hydrogen, nitrogen, ammonia, carbon dioxide, and they do find a lot of amino acid and nucleotide after operating this system for a few weeks. So the early Earth may be first hot, and then may be devoid of oxygen, but have those small molecules, inorganic molecules. And so scientists think that heat tolerant heat that is thermophilic prokaryote may reside in this environment. Okay, so people get some stromatolite. That is border fossil. This border or fossil has wavy forms of bacterial sediment, hundreds of lines of sediment. So this is sedimentary rock formed by filamentous bacteria. And so each year, when the bacteria died, and they formed the layer of rock, and then this layer of rock will be covered by another living layer of bacteria. So by hundreds of thousands of millions of years of growth, this bacterial population became fossilized and became a border, like a pillow, by the seaside, by the coast. And some people will, that is, the masons will cut it out, and they grind it into various utensils, grind into such as making lion, carving into a lion in Peking. So these are the the fossils of early bacteria. So these are the oldest ones is about 3.5 billion years old. So scientists think that of course bacteria may be first appear very old, and the second, the animals and plants may appear only about 3 billion years later than this. That is the Cambrian explosion of Paleozoic era is about 500 million years ago. Okay, this hypothesis of the early formation of the microbes, as well as the appearance of cyanobacteria, which will evolve, and they release oxygen into the atmosphere. All these was built into the theory of microbial evolution. That is, they are able to oxygenize the atmosphere and gradually make the atmosphere more than 20% oxygen. And the energy is presumed, hypothesized to be the hydrogen energy production. That is sulfide and hydrogen sulfide may form by organic reaction, not organic, it's a chemical reaction to produce hydrogen. This hydrogen may be used by primitive hydrogenase may be pumped in as electrons, and these electrons may be able to generate ATP, that is using the primitive ATPase, that is using this hydrogen energetics and the early energy producing mechanism, maybe through this hydrogen. So all these hypotheses proposed to explain the several observations. That is, we see many thermophilic bacteria, also thermophilic archaea in phylogenetically very early or very primitive bacteria and archaea. And second, many of these bacteria uses hydrogen as their energy source. So these are the hypothesis of the energy autotrophy. Energy autotrophy means that the first energy used may not be from the sun. It is from the hydrogen. Okay, so this is a summary of the hypothesis about the bacterial evolution. That is, the formation of the Earth is followed by generation of an atmosphere as a generation of sedimentary rocks. And during this period, it's called chemical evolution, or called the prebiotic evolution. The main thing happens is the synthesis of biomolecules, and these biomolecules then gradually ushered in the evolution of the cell. And then during this period, that is from before 3 billion years ago, then it forms the cell, the first cell. And then sometimes around 3 billion years ago, the cyanobacteria, that is photosynthesis, generated oxygen. And this oxygen gradually increased in its concentration. Then around the Paleozoic era, metazoan is able to form. Metazoa is a multi-celled animals. It's called the metazoan. Metazoan needs a lot of oxygen in order to provide energy. Only a high concentration oxygen in the atmosphere is able to provide oxygen, provide energy for the metazoans. So metazoans appeared about 700 or 600 million years ago. Then the Precambrian appeared here. That is around 543 million years ago. And then Precambrian, starting from Precambrian, we see lots of animal form appeared. So this is a grand scheme of the microbial evolution. So the purposes of this section is for you to organize our thinking that how bacteria may be evolved and how this bacteria is related to plants and also to animals. Okay. Then in the grand scheme of hypothesis, we have the RNA world. RNA world means that the RNA may appear earlier than DNA, maybe earlier than the cellular form. So this RNA, this stage of the evolution is called the RNA world. And then gradually it was enclosed by a membrane, and this membrane encloses proteins and RNA, and then finally the DNA. So DNA seems to appear later than the RNA. And then another hypothesis, the symbiotic endosymbiotic theory, that is the certain kind of bacteria which is getting into the larger cells and is able to form the mitochondria. This is mitochondria. And another form of the photosynthetic bacteria, which is maybe like cyanobacteria, or maybe like the Prochlorococcus. This form, it's currently not very sure about which form of bacteria is the origin of chloroplast. However, some are able to get into the larger cells, and these will be the progenitor of plants. And the cells which do not have the chloroplast, it became the progenitor of protozoa and the animals. So these are the current thinking of the bacterial evolution. So although it has not much evidence about the whole process, but the interesting thing is that it can generate, can produce a thinking to look for the sediments, to look for the fossils, to design experiment, that is a laboratory experiment, in order to support or falsify the hypothesis. That is, the whole process of scientific investigation can proceed, can go on, because we have a hypothesis. So this is very important, especially for you, the science students, because we are, as scientists, has to generate hypotheses in order to work. Okay, let me introduce a little bit of the microbial ecology. Microbial ecology. Ecology is the study of niches, the study of the living space for the living organisms. And there are many places niche, either in water or in the ocean or the base of the ocean of the deep ocean or in the soil. So let me first introduce the soil niche. The soil is very interesting because it either is produced from sedimentary material. It's called the sedimentary soil. That is such as if we go to the lake has thick sediments of mud underneath it, underneath the lake water. And when it's dried, then that became the soil. So it's not formed from the rock, it's from the sediment. It's called sedimentary organic soil. But some are formed by rock, by the weather vibration, decaying of the rock into the soil. So in mineral soil, we can see that the rock first is gradually colonized by moss, by lichens, and then bacteria, and then these produce acid to solubilize the rock, and the rock is gradually fragmented to soil, and the soil has this rock from the soil has 4 layers. That is the whole horizon, that is undecomposed plant material. A horizon as the surface soil. B horizon is called the subsoil, and the C horizon is the rock base. Is the base, is underlying rock or the fragmented rock. In this area, so we see lots of bacterial activity. So see, first it is black, that is full of organic matter, and the dark in color. It's used for agriculture, and lots of microorganism grows in here. And around the root is called the rhizome. So that space is called the rhizosphere. The rhizosphere is a place where you can find a lot of bacteria growing attached to the root systems. Okay. And but when we look at a single particle of soil, we can see that the oxygen content rapidly diminishes. So the surface is 21%. So a little bit inside of it, it became 15, then 10, 5, 1, 0. And that means that tiny soil particle, the oxygen content varies greatly. And also, in terms of its microstructure, we can see a lot of differences. We can see airspace here, and also the free water, which are held by different particles, and also the surface water will hold by the tiny particles in the soil. And we can see it's so this place has either clay or quartz, different kind of particles, different kind of inorganic material. So we can see a very heterogeneous environment for the soil environment. So we can see the microcolonies here. How do we see the microcolonies? That is, we can try to use the scanning electromicrograph to see the surface of this tiny particle. Let's see a group of bacteria. And we can also use the fluorescent dye to stain the organic material, especially the DNA. And some people is able to use the phylogenetic dye. What is phylogenetic stain? It is a small piece of RNA with a fluorescent dye. This RNA sequence is matching the 16S RNA sequence, a specific region of the 16S RNA, so that this specific dye, RNA dye, can attach to a very specific species a very specific group of species. So this is called a phylogenetic stain. Phylogenetic stain. So this is a very advanced technique to identify a group of bacteria in situ. In situ is in its original place. That is, if we see a group of microcolony here, then we want to show that they are streptomyces, and we use streptomyces dye to identify them. So these are the. So from this diagram, we can see that there are many microenvironments in the soil. So soil can harbor many different kinds of microorganisms, many species of microorganisms. That's why people thought there are 10,000 species, right? 10,000 species of bacteria in 1 gram of tropical earth, tropical soil. So that was the heterogeneity of microbes in the soil. In the liquid aquatic environment, we can see this thing which is called biofilm. Biofilm means a group of bacteria, maybe of the same, mostly of the same species or mixed species, then may form a film on the surface in the aqueous environment. That is on the rock, in the river, or in a pipe, on the human, or even in the human organs, such as in human blood vessels, on the teeth, on the surface of the teeth, in the nasal cavities. All these places, it is wet, has some water going through it, and bacteria may attach to form a colony. This colony is called a biofilm. Biofilm, a film of organic material, a film of bacteria, biofilm. The study of biofilm is very important because first, in hospitals, it will cause hospital transmitted infections to the patient. It may get into the heart duct, the prosthetics, or those kind of medical device in the human body. If those are infected with biofilms, then the cell may go into the bloodstream and may cause bacterial shock, which can cause the death of the patient. And so it's very important to study them. Currently, the study of biofilm has focused on how these one bacterial can aggregate into a colony, and this colony may form chambers, may form channels in it. And the process, one of the process called the quorum sensing. Quorum sensing, quorum, Q-U-A-R-U-M, quorum means voicing. Make your election, make your vote by voicing, make your voice loud. That is in an assembly. If there's a proposal saying that, do you agree to participate in a war? And then people will say, yes, yes, yes. And then if we hear a huge noise, a huge roar, then the chairman say, yes, we'll go to the war. So this is called the quorum sensing. Quorum sensing, that is voting by sound, voting by yelling. And this quorum sensing in bacteria is that bacteria may send out a small molecule. Small molecule. This molecule will cause bacteria to aggregate together, may cause them to swim towards these chemicals. And once they get this chemical signal, they will also send out these chemicals and this chemical to recruit other members of the same species. So they cooperatively form this slime layer. This slime will help them to stay there, will help them to defend themselves, will help them to grow into a big society. So these are the benefits of forming a biofilm on the aqueous surface. So in the water pipes, in the city water pipes, that is for the tap water, it may be contaminated with these slime layers. And also in many places, such as underneath the boat or in the industrial piping. So there are many places. If they were invaded by these bacteria, they may form biofilms. The biofilm may be very thick, so as to block the duct, block the tubing from working. So this is a very interesting area for the study. Also, an interesting place for bacteria to grow. So these are called the biofilms. You can see the bacteria in this biofilm. Okay. Another important niche for the bacteria is the lake. It's a lake system. The lake may form thermoclines. Thermocline, that is stratified in terms of temperature. That is, okay, see the temperature, you see? The temperature here. That means that from the surface, temperature may be high, and then suddenly it falls sharply, and going to the deep water, and they may stay in a colder temperature up to the sediment. So this is called the temperature stratification. Stratify means forming different layers. Okay, this stratified temperature is stable, especially in the summer. Because summer the sun shines on the surface, and so the surface is heated. But the bottom is not heated. So this stratification is very stable. But in the fall, the temperature goes down, and then the upper water and the deeper water may intermix. Okay. When it forms temperature stratification, we can see this change area is called a thermocline. That is the area where temperature falls down or rise steeply. And then the area of water above it is called the epilineum, surface of the lake. Hypolineum is the bottom of the lake. We can see the oxygen is lower in here, but the hydrogen sulfide is higher in here. So we can see that the sediment or the bottom of the lake is very different from the upper region of the lake. So in this place, we have very little oxygen, and so bacteria is anaerobic. So if they have photosynthesis, then it's called anoxygenic photosynthesis. And then in the sediment, because there is no light, so the bacteria there must be eutrophic. That is, they use all sorts of organic material for their energy source. And so this place is also more reducing because it has no oxygen. So this reducing environment may allow cells to use inorganic material for their electron receptors. So these are the environment in lakes, large or small, and sometimes in stagnant rivers. So they are all of such kind of conditions. But here we have some concepts for the bacterial ecology. That is in the sediment, and we have all sorts of guilds. Guilds means that a population of bacteria which have related metabolic activity, such as we have the number one guild called the methanogenic. Methanogenic means that the bacteria will produce methane from carbon dioxide. Or homoacetogenic bacteria, that is, they may be able to generate acetate from the carbon monoxide, carbon dioxide. That is, they can reduce this highly oxidized carbon to organic material. Why do they have to reduce it? Because they get the energy from other organic material, but they have to dump the electron back into these acceptors. This CO2 is sulfate reducing. That is, they reduce the sulfate back to sulfide, hydrogen sulfide. Okay. And also denitrifying. That is, they can use the nitrate to reduce them. Or they can do fermentation. So there are at least 4 guilds, there's 4 kinds of profession for bacteria in this sediment. But for the bacteria in the water, in the floating zone, they have the light and they have the oxygen. So they do the oxygenic photosynthesis. And the oxygen zone, that is, if there is oxygen, then for the heterotrophs, they can use the oxygen to oxidize the organic material. So this community can go near the sediments, and for this region, it has to have light. That is, a lighted area. Lighted region has to be shorter than the Arctic zones. And in the ocean, the situation is different because in the ocean there is ocean flows, so the oxygen is more abundant at the ocean bottom. That is, the ocean bottom is not anaerobic. On the contrary, it may be aerobic, quite aerobic. So sharks or large animals can dive into the bottom of the ocean. Okay, so this is more detailed explanation of the aquatic environment. In the river, it's different. In the river, the standard situation is like this. That is, if we have a river, but the river is as an input by sewage or the waste material. So this is very easy to see because as long as there are human beings living around by the river, we'll dump our wastewater into the river. So upon the dumping, we can see what happens. Once we dump organic material into the river, then we can see this red curve. That is, bacterial greatly proliferates, and the organic carbon is also input into the river. Biochemical oxygen demand means that the oxygen is highly demanded for bacteria to oxidize the organic carbon, right? So the oxygen demand is high, so as to cause the oxygen to fall down, to go down dramatically. And in this process, a lot of bacteria grows up, and somewhere downstream, when oxygen start to rise a little bit more, then algae and the cyanobacteria can grow. That is, algae cannot even grow in this place where oxygen is not available, but they can grow when oxygen started to recover, because the cyanobacteria will release oxygen into the water. So we can see a lot of green growth in this area by algae and cyanobacteria. But in this place, we see a lot of dark-colored bacteria growing in this region. And gradually, oxygen will be produced by the algae and the cyanobacteria. And then we can see, that ammonia is generated. Ammonia phosphate is also released because the phosphate is abundant in our detergent. So when we use water, we use the machine to wash, the detergent is full of phosphate, and also ammonia is produced from the organic material. And so we can see that ammonia and phosphate is in here, and the nitrate is in here. So both of them reaches peak in here. So this is a general description of the environment in the river, which were polluted by sewage from human residents. This is a picture of the place where the water is heavily polluted by algae and the cyanobacteria. So this looks like a lake, a heavily contaminated lake. So this is also called eutrophication. Eutrophication, because a lot of organic material, phosphate, nitrogen, is from sewage, which was released into the lake. And the lake has so much organic material, so it's called eutrophication. Eutrophication will cause a lot of algae and cyanobacteria that is very hostile to large animals such as fish or aquatic mammals to live. So this is a general outline of the river ecosystem for bacteria. And I also introduce some about the black, that is the ocean bottom ecology. Ocean bottom, they are the black smokers or white smokers. Black smoker, white smokers at the bottom of the ocean. This was discovered, I think, in the 1950s, when people are able to build a submersible submarine, and people will get into it, and they go down to a depth of 3,000, 4,000, 5,000 meters underneath the surface, and they observe these black smokers and these white smokers. And the chemical process is this: the seawater goes down to the ocean bottom, and the ocean bottom was heated by the heat there, and so they will be sometimes reduced. These chemicals reduced to this kind of more reduced form of the minerals. And then in the place where the water comes out, then it will be oxidized to the black color. So this is called the black smoker. The hottest temperature can be as high as 350 degrees Celsius. When temperature is very low, is lower, that is under 100, and so it became a white cloud. So it's called a white smoker, the black smoker. So these are the places which we can see many strange animals and also strange archaeas in here. Okay, let me explain the geochemical cycles. In ecology, we are concerned with the cycling of the elements. The first is carbon. We can see that carbon is is cycled here. First is the rock formation is forming the fossil fuels, and people get fossil such as the petroleum and the natural gas, natural gas from the rock, from the rocks, and then by burning them, release back into the atmosphere. And the land plant will absorb this carbon dioxide, and then we fix them into carbon, organic carbon, will be used by animals and microorganisms. And in the ocean, carbon dioxide is dissolved in the ocean, which provides the carbon dioxide for the aquatic plants. And these aquatic plants were eaten by the aquatic animals. And these dead animals and dead microorganisms were from the sediment, or became the coal, petroleum, as well as the natural gases, which will be recovered by human activity. So these are the carbon cycle, which I think you will be very familiar with. And so we can see the process, which is called chemolithotrophy. That is eating the inorganic carbon for the nutrient, chemolithotrophy. And the processes here are the oxygenic photosynthesis here, then anaxygenic photosynthesis underneath the cycle. Here, methanogenesis. Methanogenesis is forming the methane from the methyl compounds. But methane is eaten by bacteria, which called the methanotrophy, eating the methane. So there are bacteria which are specializing in eating methane. It's called methanotrophic bacteria. So we can see that the carbon is cycles in the bacteria in these few cycles. As for the nitrogen, you can see that there are many processes. Nitrogen fixation is for the atmospheric nitrogen to be fixed into ammonia. This ammonia can be assimilated to the amino group in the proteins. Okay, and then ammonia is oxidized to the nitrate form, and this can be then reduced to NO, N2O, and reduced to N2, and so these will be released into the atmosphere. So we can see nitrogen, they can be fixed and then used in the organic in the organisms, and then can also be oxidized through these different forms, and then reduced back into the nitrogen. So these are the natural cycle in the biosphere. Under the sulfur cycle, the nitrous cycle. So these are the bacteria for the nitrous, the nitrogen cycling. It is bacterium, which I have mentioned before. And these are the nitrogen fixing bacteria, which I have also mentioned before. Okay. And finally, I mentioned the rumen ecosystem. Rumen is a system where we can, where the cattle and the goat and the camels, they will eat the grass. The grass, and the grass will first get into the reticulum, and sometimes will be regurgitated for the animal to chew into finer pieces, and then go and was swallowed again into reticulum. Then go into the rumen. Rumen was fermented. So this is a big fermenting jar, fermenting vat, which is about 200 liters for a cow. After fermentation, lots of bacteria grows in it. Then it will then be sent into here. Be sent into this omasum, and then into the abomasum. In the omasum and the abomasum, especially the abomasum, it is like the human stomach. That is where a lot of bacteria were used by the animal, where the chemicals formed was absorbed in the small intestine. So these four chambers of the ruminous animal is a place of lots of bacteria where they can digest the cellulose, they can digest a lot of material which are not digestible by mammalian animals. So these are the window into the rumen. And we can see lots of sugar and the cellulose was digested into smaller fatty acids, which are used by the ruminant animals, and also produce lots of vitamins, these kind of things for the animals to use, and also carbon dioxide, carbon methane was it was eructation, eructed to atmosphere. Eructation means that belch it out into the atmosphere. So that's why cows can belch out the methane, the marsh gas into the air. Okay, so let's have 10 minutes rest. 10 minutes rest. So we start at 27.